Hello student. Okay, so in today's class we will go through few questions as a tutorial session that will cover the lecture of chapter 1. If you remember, in chapter 1 we established that the innate immune system is your body immediate hardwired defense. It's always on, it's always ready, it requires no prior exposure to a pathogen to function, contrarily to the acquired immune system. We explored how it detects danger through pattern recognition receptors, particularly the toll-like receptors, and we listed many of them with their location, their function, which sits on cell surface, to summarize, and in endosomal membrane, and recognize conserved microbial structure that we called pathogen-associated molecular pattern, PAMS. If you remember, we mentioned it a lot of time. So as well as the host derived danger signal that we also mentioned, that is called DAMS, the damage-associated molecular patterns. So we then examined how the system responds to danger, to infection, through the complement cascade, through the cellular machinery of neutrophils and macrophages, through the vascular and the cellular events of acute inflammation, so and also through phagocytosis and intracellular killing. So every one of those mechanisms will be appear or will be in the center of one of the questions of this tutorial. So just please read the lecture note first and come back to try to solve each of the question on your own first, and then listen to the answer to find out if you have it you have it right. Okay, so let's continue with the first question. Okay, we will go through 21 questions for this multiple choice questions type, and for each of them I will first select the right answer and explain also why the others are not correct answers. So the first one is, which of the following best describe a defining feature of the innate immune system? So we have A, it generates immunological memory after each infection. Its receptors are encoded in germline and do not require somatic recombination. It is activated only after the adaptive immune system fails, or it relies exclusively on lymphocytes for pathogen elimination. So the correct answer is answer B. Its receptors are encoded in germline and do not require somatic recombination. So why the answer A is wrong? Because it generates, we are talking about memory, immunological memories. When we are talking about memory, it's referred here to adaptive immunity. It's a hallmark of adaptive immunity. As we know, B and T cells is not a characteristic of the innate immune system. C is also wrong because the innate system is the first responder. Please do not wait for adaptive immunity to fail before coming in, so it acts immediately. And D is also wrong because lymphocytes belong to the adaptive system. It doesn't belong to innate cells. So innate cells either include neutrophil, macrophages, natural killer cells, and dendritic cells. So lymphocytes belongs to adaptive system. Okay, for the question 2, pathogen associated molecular patterns, PAMS, are best described as A, molecules unique to each individual pathogen strain. B, host-derived danger signals released during tissue injury. C, conserved molecular structure shared across broad class of microorganisms. And D, antibody fragment generated during infection. The correct answer is answer C. We are talking here, PAMS are best described as conserved molecular structure shared across broad class of microorganisms. when we identify, if you remember during the lecture, we identify PAMS has a signature, a kind of pathogen signature, an unique identifier for the whole microorganism branch. So like we identify example like lipopolysaccharide or flagellin or peptidoglycan that are structural features essential for microbial survival, and therefore they are conserved across entire pathogen groups, and they are not unique to individual strain. Okay, and yeah, so A is not correct, because if they were unique to each strain, if PAMs were unique, then we will have a large panel of them. This is more like antibody synthesis. Okay, so B is wrong, because it describe we are talking here about tissue injuries, so it's refer more to damage, to damage associated molecular patterns, not PAMs. And these ones are derived from host danger signal, not microbial molecules. Okay, and the answer D is wrong, because we are talking about antibodies. So once we are talking about antibody, from the moment you read antibodies, this is adaptive immunity. We are not in immune immunity, when your pump are key component of innate immunity. So the right answer was C. Is the answer C. The third question: which of the following is an example of damp rather than a pump? So we explained already. We have lipopolysaccharide that are normal constituent of the gram-negative bacterial outer membrane. So it's classic pump. And flagellin, we also explained flagellin is a structural proteins belonging to the bacterial flagella. So it's normal component of bacteria. It's a classical pump. And for HMGB1 released from necrotic cells, the answer C is the right answer, because from the word necrotic cells, you can notice there is a damage here. So it's more indicative to damp than pump, the necrotic cells. And the last one, peptidoglycan from gram-positive bacteria, it's natural component of the cell wall of bacteria, so it's a classical pump. So the right answer was C. The meaning of HMGB1 is high mobility group box 1. It is a nuclear proteins released by damaged or dying host cells. So it's a damp. Okay. The fourth question: toll-like receptors signal through which of the following principal adapter proteins? So you have four couple of proteins, and to find out the right answer, we need to know the role of each of them, of these two pairs. First, the right answer is A, TRIF and MyD88, because these two main adapter, there are two main adapter actually of downstreams of TLR activation. So the first one will drive type 1 interferon production, when the second one will drive pro-inflammatory cytokine. This is the hallmark of innate immunity, using passing by toll-like receptors. So for the second pair, RAC1 and RAC2, they are enzymes that catalyze VDG recombination in lymphocyte. So it completely belongs to the adaptive immunity. So the CpG DNA motif, I think they are used for antigen presentation, so they are not innate. They don't belong to the innate system signaling. And D, the lipopolysaccharide. No, I mean I am reading from question 5. Sorry. Type 1 and type 2. Actually, they are transport peptide, and they are using the antigen presentation, so they are not innate signaling. Okay, not CpG. That was I was confused a bit. So, and the proposition C, type 1 and type 2, they are involved in the antigen presentation, so this is more innate signaling. And for D, the couple SIC and CARD G9, they are signaling molecules downstream of C-type lectin receptors, not toll-like receptors. Okay, so the right answer is A. The question 5, toll-like receptor 4. Now we are focused on one of them. Toll-like receptor 4, primarily responsible for recognition, recognizing which of the following pump. We have viral double-stranded RNA, bacterial flagellin, CpG DNA motif, and lipopolysaccharide from gram-negative bacteria. So the right answer is D, LPS from gram-negative bacteria, because toll-like receptor 4 is the canonical LPS receptors. So yeah, it's for A. Why the answer A is not okay? Because the viral double-stranded RNA is detected by TLR3, that is endosomal located, and cytosolic RIC1 MDA5. So these are the toll-like receptors particularly involved in the double-stranded RNA, viral double-stranded RNA. For B, the answer B is also wrong. Flagellin is recognized by TL5, that is located on the cell surface, and CpG DNA is recognized by TLR9, that is located in endosomes. So the last question of this slide, which TLR are located on intracellular endosomal membrane? Which TLR are located on intracellular endosomal membrane? So we have for position 1, TLR 1, 2, and 4, TLR 3, 7, and 9, TLR 5, 6, 10, and TLR 2, 5, 9. So the right answer is B, 3, 7, 9, because these ones are inside the cell in endosomes, because also they are ligands. The viral nucleic acid are only accessible after the pathogen has been internalized and degraded. So the answer A is wrong. The couple 1, 2, and 4, or the triple 1, 2, and 4 are surface receptor, so they are detecting either bacterial component. The answer C is also not okay, because TLR5 detects flagellin, and TLR6 lipoprotein, and they are also with TLR2, they are surface receptors, so they have particular targets, flagellin and lipoproteins. Okay, the answer D is not okay, because TLR2 is a surface receptor mixing surface and endosomal toll-like receptors in one answer, make it incorrect. Yeah, because here we have a different target, even if you have one that is okay, but yeah, since you have 5 and 2 that are not targeting the same, so the answer will be wrong. Okay, so the right answer is B, 3, 7, and 9. Good. Okay, question 7. Activation of TLR ultimately drive expression of pro-inflammatory cytokine, primarily through which transcription factors? We have GATA3, FOXP3, NF-kappa B, and TBET. So the right answer here is NF-kappa B. NF-kappa B for nuclear factor kappa B is the master transcription factors for inflammation. So it's well known of the factor for inflammation, and it drives gene encoding for TNF alpha, for interleukin 1, interleukin 6, and interleukin 8, and many other inflammatory mediators. So why answer A is wrong because GATA3 is a transcriptor factor for Th2 helper cells differentiation, so it's adaptive immunity. FOXP3 from the answer B is also for regulatory T cells for Tregs, so it's adaptive immunity. And in the answer D, the TBET, the drive Th1 T helper cells differentiation, so it's still adaptive immunity. So finally, we have only C that belongs to innate immunity. Okay, so for question 8, the NLRP3 inflammasome upon activation leads to the maturation and secretion of which cytokines? We have 4 couple. The first one, A, EL4 and EL13, EL1 beta and EL18, interferon alpha and interferon gamma, TNF alpha and EL6. So which one is the right answer? The right answer is B, EL1 beta and EL18, because NLP3 assembly activate Caspase-1, which cleave the inactive precursor pro EL1 beta and pro EL18 into their active secreted forms. So this cytokine drive fever and inflammation. These two particularly are working in the drive inflammation. Okay, so the answer A, EL4 and EL13, they are Th2 cytokine. They are involved in allergic response and anti-helminth immunity. So there's no connection with inflammasome. And the answer C, interferon gamma and interferon beta, they are type 1 interferons, so they are produced through TLR7 and TLR9 signaling pathway, not the inflammasome. So, and for D, it's also wrong because TNF alpha and EL6 are produced via the nuclear factors kappa B downstream of toll-like receptor signaling, not using Caspase-1 inflammasome activation. Okay, so the answer is D. And for the question 9, RIG and MDA5 primarily detect. Answer is C, viral RNA in cytoplasm of infected cells. So why is the right answer? Because these RIG1 and MDA5 are cytosolic RNA helicase, so they patrol the cytoplasm for viral RNA signature, and their activation drive type 1 interferon production. The first, bacterial lipoproteins, is wrong because bacterial lipoproteins are detected by surface TLR2 and they are not cytosolic. Fungal beta-glucan, they are also detected by lectin-1. This is a C-type lectin receptor that is located on the surface. D is wrong because D belongs to the extracellular complement fragment. They are soluble plasma protein detected by complement receptor. This has nothing to do with intracellular viral sensing. So next questions. 10. Which C-type lectin receptor play a critical role in recognizing beta-glucan on fungal cell walls? TLR2, lectin-1, MDA5, or node 2? The right answer is B, lectin-1, because lectin-1 is the primary innate receptor for beta-1 triglycan. This is a major component of fungal cell walls. So lectin-1 is the primary innate receptor to detect fungal cell walls component. And TLR2 recognize fungal component too, but it's not a C-type lectin. So here the indication of C-type lectin will differentiate the answers. And it will not specifically target B glucan like lectin-1 that is specifically targeting the glucan. So for C is wrong because MDA5 is a cytosolic RNA helicase sensing viral RNA, is completely unrelated to fungal recognition. and D is wrong because NDO is cytosolic NLR receptor recognizing bacterial Muhammad dipeptide from peptidoglycan, not fungal structure. Okay. Question 11. The complement system achieves which of the following three principal effector functions? We have antigen presentation, cytokine secretion and phagocytosis, opsonization, direct lysis and inflammation. We have T cell activation, B cell activation and natural killer cell killing, also somatic hypermutation, class switching and memory formation. The right answer is B, opsonization, direct lysis and inflammation. These are the three core roles of complement activation. So C3b coats pathogen for phagocytosis or for opsonization, and the C5b to C9 to the bunch of C9 will punch holes in the membrane while forming the membrane attacked complex that will induce the lysis of the cells, and the C3E and C5A will recruit C3A actually and C5A will recruit and activate inflammatory cells. So we are for the opsonization, then direct lysis, then inflammation. Okay. A is wrong because their antigen presentation and cytokine secretion are function of dendritic cells and macrophages, not complement protein. This belongs to dendritic cells and macrophages, not complement protein. And this answer C is wrong because T cells, B cells, and natural killer cells activation are adaptative. They are coming from adaptive immune system. So the complement will amplify but does not directly perform these kind of functions. And for D, the somatic hypermutation, a class switching, and the memory formation, they are B cell process in the germinal center, so they belong to adaptive immunity. Question 12. Which complement pathway is activated by mannose binding lectin, MBL, binding to carbohydrate on microbial surface? We have classical pathway, alternative pathway, lectin pathway, and terminal pathway. Yeah, if you read your note very well, you already know that there is no terminal pathway. Actually, there are only three pathways forming the complement system: the classical, alternative, and the lectin. So which complement system is activated by mannose binding lectin? Is the answer is C, lectin pathway. Mannose binding lectin is actually MBL is defining recognition molecule that the lectin pathway. So surely when you see mannose binding lectin, is lectin pathway that is involved in it. So why A is wrong? Because classical pathway is activated by C1q. That also is triggered by the binding of an antigen and antibody complex. So classical pathway refer always to the formation of antigen antibody complex, and alternative pathway will refer to, we start with no recognition molecule. Actually it's alternative. That means it can be chemical, it can be from a disturbed activation or functioning of the pathway, but not really pathogen involved in it. So there is no recognition of molecules like we can have with MBL. For the answer D, we said already, there is no separate completely pathway that is called terminal pathway, but we have this terminal component of the three pathways. They are sharing the same terminal component. So it could be a sub pathway, but it's not really a pathway, that one. And this terminal route is shared by all the three pathway, classical, alternative, and lectin. Okay. 13. The alternative complement pathway is initiated by antigen antibody complex formation, MBL binding to mannose residue, spontaneous low level hydrolysis of C3 takeover, or CRP binding to bacterial phosphocholine. The right answer is C, spontaneous low level hydrolysis of C3, because C3 is unstable and continuously hydrolyzed at a low rate in plasma, generating C3H2O. So answer A is wrong, because antigen antibody complex formation will initiate the classical pathway. Yeah, not the alternative. And B is wrong, because MBL, we say that in the past questions, is lectin pathway, so also wrong. And the D, the CRP, C-reactive protein binding to bacterial phosphocholine, will activate the classical pathway by recruiting C1q. So it's not alternative pathway either. So in each of them, we have the responsible agent for activation that is different, and that will help us to identify which pathway actually is involved, so that we can refer the one that is that correspond to alternative complement pathway, like here. And 14. C3b is a complement fragment that primarily functions as a chemotactic signal for neutrophil recruitment, an opsonin enhancing phagocytosis, a component of the membrane attack complex, or a vasoactive amine increasing vascular permeability. The right answer is B, an opsonin enhancing phagocytosis. Yeah, C3b attached to pathogen surface, and it's recognized by complement receptors on macrophage or neutrophile, so it will dramatically increase the phagocytic uptake. So it will induce opsonization. The C3b referred to opsonization. So it's a complement fragment that primarily functions as an opsonin enhancing phagocytosis. A is not okay, because C3a and C5a are the one that are anaphylotoxin that act as chemotactic. Chemotactic signal, when we talk about chemotactic signal, we think about C3a and C5a, not C3b. And the C is wrong, because here is C5b that will initiate the formation of membrane attack complex. A component of the membrane attack complex here will be C5b, not C3b. And why D is also wrong, because we are talking about vasoactive amine increasing vascular permeability. This is the role of histamine and serotonin that will increase vascular permeability, not C3b. C3b don't play this role. For question 15, which complement fragment are referred to as anaphylotoxin due to their pro-inflammatory activities? We have C1q and C4b, C3b and C5b. We have C3a and C5a, and C2a and C4a. I think we said that already in the previous question. The answer is C, C3a and C5a, because they are the small peptide fragment cleaved from C3 and C5, and they bind to receptor on mast cells, on basophil, on leukocyte, and they will trigger the degranulation of histamine that will increase vascular permeability and increase also anaphylotoxin income. Okay, so A is not is wrong, because C1q and C4b. C1q is a recognition molecule, C4b is a part of C3 converter, so neither has the anaphylatoxin activities. And for B, it's wrong because C3b is an opsonin, and C5b initiates the membrane attack complex, the assembly of the assembly of the membrane attack complex. So it's not C3b and C5b. And D is also wrong because C2a and C4a are generated during classical and lectin pathway activation. C4a has a very weak anaphylatoxin activity, so the best choice is C3a and C5a. Okay, next questions. The membrane attack complex is formed by which complement component? We have C1, C2, C3, C3b, factor B, factor D, C5b, C6, C7, C8, and C9, and the last one C4b, C2a, and C2b. Yeah, that I think that one all the class will find out the result because we explain it and we draw it. So the membrane attack complex is constituted of C5b, C6, C7, C8, and C9. That is the right answer. After the C5, the cleavage of C5, we have C5b that will remain on the surface, and it will sequentially recruit 6, 7, 8, and recruit many of the C9 molecules that will form a poly C9 polymerase, and they all combined will form a transmembrane pore. So the pore will go all along through the membrane, and this will cause osmotic lysis of the cell. So A is wrong because C1, C2, and C3, they are early activation components. They generate the converters, but they do not form MAC. Membrane attack complex. Answer B is also wrong. C3b, factor B, factor D, they form the alternative pathway C3 converters. And C4b, C2a, and C3b, they form the classical and lectin pathway C5 converters. So the right answer is C. Question 17. CD55, also called DAF, and CD59 protect host cells from complement-mediated damage by degrading C3b and C4b on cell surface, preventing MAC formation and complement deposition on cells, blocking TLR signaling on host cells, or producing anti-inflammatory cytokine. So the right answer is B, by preventing MAC formation and complement deposition on cells. So the CD55 will accelerate breakdown of C3 converters, and the CD59 will block the final step of membrane attack complex assembly by preventing the formation or the insertion of the poly C9. So why A is the wrong answer? Because the factor 1 is the serum protease that will cleave C3b and C4b. So it's not the same mechanism. C is wrong also because CD55 and CD59 have no role in blocking toll-like receptor signaling. So this is a completely different pathway. And the D is also wrong because anti-inflammatory cytokine production is performed by cells like Tregs and M2 macrophages, not by membrane complement regulatory proteins. Question 18. Which of the following is the correct sequence of events in leukocyte recruitment during acute inflammation? The correct sequence is B, the rolling, activation, and firm adhesion in diabetes, because this is a strict order of process. So we have leukocyte first, that will slow down the progression and roll. The rolling will be mediated by selectins before the chemokine will be able to activate integrin, which then will mediate firm arrest, after which the cells squeeze through the endothelium using diapetesis. Okay, so the order are not correct. This is the right order, is strict order of process in the inflammation. Okay, so 19, the initial rolling of leukocyte on activated endothelium is mediated by interaction between, answer is B, selectin on endothelium and carbohydrate ligand on leukocyte. Yeah, it's the selectin on endothelium. This will the activated endothelium will rapidly express selectin, and this will bind and release carbohydrate structure that are on the surface of leukocyte. This will create a weak reversible interaction that will cause the rolling. So sorry. So A is wrong because integrin binding sorry. Okay, we will start with exercise 1, the clinical scenario. So when we read a clinical scenario in our future practice, in our future exercise, please resist to the temptation of jumping directly to the diagnostic, even if you have, because sometimes when we can read keywords that will indicate something that we recognize in the immune system pathway, we want to directly jump to the diagnostic, but instead we should walk through it in the layers. So read every single sentences of the scenario 3 times. Start with the patient infectious history, which organisms are causing the problem and which are not. Determine what are the causes and what is actually is not related to any infection or pathology, and that pattern is your first clue about which arm of the immunity is affected. Then you look at the laboratory data, what is present, what is absent, what is reduced, what is increased. So read carefully the laboratory data and map those findings onto the immunological component you know. So make each of the data to correspond to a immunological component that you know. Then ask, does the mechanism fit the clinical picture? If the whole picture actually is clear, is logical, is understandable. And finally, what would happen if you corrected the defect and what remained unaddressed? Think about all these questions before really jumping into the clinical scenario answers, starting answering all the questions. So the logical sequence, this logical sequence that will really guide you, and it will be good actually to use it for every clinical case in the next exercise. So let's read this first clinical scenario. A 28-year-old man presents to the immunology clinic with a history of recurrent severe infection with Neisseria meningitis and Streptococcus pneumonia since childhood. He has no history of viral infection more severe than those of the general population. Laboratory investigation revealed markedly reduced serum level of C3 with indetectable C5, C6, C7, C8, and C9. The neutrophil count, T cell subset, and immunoglobulin levels are all within normal limits. So this is the description. Question now for discussion. First one, based on the laboratory finding, which component of the immune system are deficient in this patient? Identify the specific defect and classify it within the innate immune system. So you have, in one question you already have like 3 tasks. Which component of the immune system are deficient? Identify the specific defect, then classify it within the innate immune system. So this answer should have 3 components. Okay. Second, using your knowledge of the 3 complement activation pathway, explain why a deficiency in terminal complement component C5 to C9 render this patient specifically susceptible to Neisseria and encapsulated bacterial infection. Why are other pathogens not equally affected? Third question. So here you should use your knowledge to the second question you should use your knowledge to answer, to be able to answer. It's something that is like your critical thinking. You have to use your logic, to use the prerequisite knowledge that you know already to answer this question. So make the theory to fit with the practice. And in the third question, the patient neutrophil count are normal. Here you have a laboratory result. Neutrophil count are normal. Explain why normal neutrophil function alone is insufficient to compensate for complement deficiency in defense against this organism. So why this normal neutrophil function alone is insufficient to compensate for complement deficiency in defense against this organism. In your answer, describe the role of C3b opsonization and the MAC in antibacterial defense. This is for the 3 first questions. So for the first question, let's come back to the first question. What is the immunological defect in this patient? So first, we say this patient has a terminal complement deficiency. It has a terminal complement deficiency, specifically because the absence of C5 to C9 with reduced C3. So this justifies that the terminal component that starts at the C5b, if it's absent, that we have a terminal complement deficiency. And this is a defect in the innate immunity, since the complement system belongs to the innate immune system. So this is a defect in the innate immune system. We already identified the deficiency, where which part of the immune system is defectious, and now specificity of it is humoral innate effector arm, the complement cascade. So the terminal component C5b to C9 normally is assembled to form the membrane attack complex, which complex is actually used to put a hole, to punch a hole in the bacterial membrane, causing osmotic lysis. So without this C5 to C9 protein assembled as a MAC, if there is no MAC, there cannot be any form of all the upstream activation of the complement will be useless. No membrane attack complex can be formed if there is no C5 to C9 activation, and there will be no result of this activation, no effective result, no concrete action after the activation of the whole complement pathway cascade. So if you want to answer progressively, we will say first, which component of the immune system are deficient? Is particularly the terminal complement that is deficient. Identify the specific defect. Is the innate immune system that has a defect here. If you want to classify it within the innate immune system, we will classify it in the humoral innate effectors, humoral innate response. Okay. Sorry. So for the second question, actually it was, why do this deficiency cause susceptibility specifically to Neisseria and encapsulated bacteria? Why other pathogens are not affected? So here first, what we should know is that Neisseria species are gram-negative bacteria, and they have an outer lipid layer membrane that is unique that is vulnerable to membrane attack complex, since this membrane attack complex mediates the pore formation and osmotic lysis of this type of membrane. So if there is no membrane attack complex that is formed, there will be no serum bactericidal activity against Neisseria. And this cannot be compensated by phagocytosis alone, because bacteria will replicate rapidly and in the bloodstream. So before phagocyte can contain it, it will be everywhere. And for encapsulated bacteria, they also resist to direct phagocytosis because their polysaccharide capsule blocks complement deposition, and also it's block also the pathogen recognition receptors. So in healthy individual, this membrane attack complex will provide a critical backup killing mechanism. But if there is not there, then the other pathogen will not be, the susceptibility will be focused only on those ones, since the protective mechanism will be weakened by the absence of MAC. And for other pathogens like viruses, intracellular bacteria, or fungi, they are controlled by cell-mediated immunity, by like natural killer cells, Th1 activated macrophages, so which is fully intact in this patient. In this case, they don't mention that this part of cell-mediated immunity has any damage. So the other pathogen will be controlled by this branch of innate immunity. So this will explain why his non-bacterial infection are normal when bacterial infection are not normal. The question 3: why normal neutrophil function is insufficient to compensate for complement system? There are many reasons that can justify. One of them is the phagocytic efficiency is severely reduced without opsonization that is provided by the formation of MAC complex. So if the phagocytic efficiency is weak, then neutrophil engulf bacteria most efficiently when they are coated with C3b. So without complement, the neutrophil must rely on a weak opsonic pathogen recognition receptors. So inadequate, again, encapsulated organism, because they have a physical shield on the bacterial surface from direct contact, so they will not be easy to recognize them. The MAC provides serum bactericidal activity independently of cells. Another way to answer is that the neutrophil recruitment itself is impaired. The neutrophil recruitment itself will be impaired. So C5a is one of the most potent neutrophil chemoattractant. If C5a, without the cleavage of C5 into C5a, if we don't have generation of C5a, then that means there will be less chemotactic gradient. It will be reduced, and this chemotactic gradient is supposed to guide neutrophils to the infection site. So no C5, no C5a, no chemotactic gradient or attraction, so neutrophil will not arrive to the infection site, and the efficiency of the action will be damaged or dampened. Okay, for the next question, 4. A second patient with a similar infectious history is found to have normal terminal complement but undetectable mannose binding lectin (MBL). Compare and contrast the immunological consequences of MBL deficiency versus terminal complement deficiency. Which complement pathway is specifically affected by MBL deficiency? Design a diagram illustrating the 3 complement pathway, indicating the precise point at which each of these 2 deficiency, terminal complement and MBL, interrupt the cascade, and the effector function lost as a result. So an indication for the answer 4, that is about to compare MBL deficiency versus terminal complement deficiency. So you can draw we will do that in the next class. We can draw a table with these two deficiencies, and for each of them we will indicate we will indicate what is the result or the action. But we should notice that MBL deficiency is really mild, because we still have the classical and the alternative pathway that could compensate when since MBL will refer to the lectin pathway. We have two more. The complement system have three pathways. You have classical, lectin, and alternative. From the moment you have a deficiency with the lectin, the two others can compensate a bit, but the terminal complement is that common shared pathway between the three, and it's also the effector arm of the complement system. So all the three pathways lead to that terminal complement pathway. So if there is not there, if that one is not there, none of the pathway could be effective, could give any result. So find a way to draw a table to compare these two, and you will use all the target point to indicate which complement pathway is specifically affected by. First we'll identify which complement is affected, and then compare the immunological consequences of these two. Okay, and the 5 questions is about drawing. So you will draw the three complement pathway and show where the deficiency occur. The first one, the MBL, and the terminal. So this can help you actually to answer, to do the comparison, since you will have a drawing. You can see from the line where you have the cut, where you have the insertion of this deficiency. You will know that okay, if it stop here, if it start here, that means the remaining part will be affected or will malfunction. So you can have idea how to use that to indicate what will be the immunological consequences. Okay, so for the next class we will discuss this together and see the different correction on proposition of the student on these two answers. Okay, thank you, and see you for the next tutorials. Bye.